Hi everyone, welcome to Rice. I hope you've been having a good week so far. My name is Dr. Lan Lee, my pronouns are they, them, and I'm in the history department here, and I also work in the medical humanities program, and I teach the Intro to Medical Humanities class. So in terms of what I'd like to talk about today is a little bit about when I was in undergrad, when I started college, I had no idea what I was going to do, but most of what I work in now is between science, medicine, and the humanities. So that's really what I'd like to think through with you in terms of what you'd like to get out of Rice during your time here and what you'd like to do with your life beyond Rice. So my case study really is a little bit of myself in terms of having few role models who were doing this kind of integration work, because there isn't that much space to think between sciences and the humanities beyond a liberal arts education. And so what does that look like? And specifically, when I talk about science, medicine, and the humanities, I don't mean this kind of, there weren't really programs that led me to where I am now, but I started off as a bio major, I became a history major, and then was also working in film, and I also produced a lot of podcasts and art pieces, and I'll tell you a little bit about how all that comes together in my own work now. So this starts back to, this is me, there I am. In terms of my own background of where I grew up, life is not linear, but here is a linear timeline. And in terms of the beginning of life, I was born. And my parents are musicians. They're Chinese Conservatory graduates. They specialize in Chinese music. My dad's a classicist. And I played a lot of music growing up. I did a lot of guzheng, a lot of dulcimers, and I was always playing with percussion and with all sorts of sounds, being thrown on stage and learning Chinese opera kunqu in 2 weeks. So there was a lot of improvisation in my own life growing up. But because my parents were artists, they always told me to stay away from the arts. So of course, when I started college at Barnard, I decided to pursue science. I was like, maybe bio is the way to go because it's what I should be doing. So there's this narrative of what you should be doing versus what you are able to do and what you love to do. So I thought I should do science. I worked in an HIV lab, and I ended up teaching bio as a supplementary instructor. And one of the main questions I had when I was working in lab were questions that I couldn't answer by being in lab. So I was, doing a lot of mini preps, and I kept on wondering, why was I doing this kind of science? Why was I focusing on HIV, and why did this research feel endless? So I had these questions in lab that I felt like, being a basic technician, I couldn't answer. So I started teaching science and then fell into the history of science. I did not want to be a history student. I was a horrible history major, or a horrible history student in high school, and then fell into becoming a history major. So I did history of science. But I was also really interested in medicine as well. So when I studied abroad at University College London, I was taking history of science classes and also exploring all the options that London had to offer. But at the same time, because I was a Pell Grant student, which meant I was a scholarship student, I was funded by the Gilman Scholarship to make video blogs. So that is when I started making film. This was in 2008, early days of YouTube. I was also making music and posting videos every week and doing this kind of self-production with my little camcorder. And it was an amazing experience. It was really, really hard, but it made me, and it trained me in that first 100 hours of my 10,000, would be 10,000 hours of becoming a filmmaker. So after graduating, I freelanced for a bit. I was working for production studios, and I was also applying to grad school. And my mom at the time was like, why aren't you applying to grad school now? And I was like, because I'm not ready yet. And I was learning a lot. I think in that period, after graduating college, I learned how to learn. So much of college was preparing for the sake of an essay, for the sake of an exam, and learning what I thought the professor wanted me to know. And once I went into the real world, I realized that there's so much to know, and there's so many ways of knowing. And that started to stimulate an inherent interest in myself of what kinds of questions I could ask and explore on my own time. So I applied to grad school, and I ended up getting into MIT in their History, Anthropology, and Science Technology Studies program, also known as HASTS. But the abbreviation that's most relevant is this term STS, this acronym STS, which stands for Science Technology Studies. And it really gave me a space to ask the questions that I wanted to ask, which was, why do we approach science, and why do we really celebrate science in the way that we do, and what kinds of questions do we leave out? So the social questions, the legal questions, the ethical questions, those weren't questions that we covered in class. And so one of the basic questions that STS presents is, what is a fact? So how do we agree on things in the world to be true? So one basic example is if you ask someone what color the sky is, they will tell you that it's blue, but the color of the sky changes throughout the day. It can become orange, it can become red, and yet our gut response is to say that it's blue because we've agreed that there's a certain wavelength that registers a certain color to us that we've labeled as blue, that we can call it as a stable category of the color of the sky. So STS kind of starts with allowing us to think about what is a fact and how do we agree on facts. STS also allows us to think through questions of bioethics. Who does science help? Who does it harm? What are the conditions of its limits? And for me, I was thinking more into the realm of medicine and biology, which was, how do we know elements about the body? How do we know how body parts work, what organs do? And this might seem like a really basic question, because if you ask someone what organs do, they can feel their heart and say, my heart is beating, my heart is meant to beat. But the body is so complicated. There's so many things, invisible things in the body that we don't really know about and that we can't quite access. So one of the main questions that I was interested in was elements of Chinese medicine, for instance, which is something that I grew up with, but I never really had the space to explore because science was universal and science was a space that excluded other ways of knowing. And so I was interested in these other ways of knowing that existed beyond the realm of the science that I was taught and the science that I loved. So in thinking about these other ways of knowing, another big question was what counted as knowledge. If a fact is socially constructed, then who created those conditions of agreement? And who excluded people or ways of knowing from that conversation? So what counts as knowledge? And also, by extension, what counts as science? And so the main topics I was interested in grad school was histories of Chinese medicine and also histories of medicine in South Asia, looking at Ayurveda. So I spent, I think 2012, 2013, making a short film about integrative medicine when I was in Mumbai, and trying to think about, again, knowledge in these other ways of knowing that, again, I had no space or opportunity to think through as a science student when I was an undergrad. And one really useful framework that I developed later that I didn't have at the time was this framework of decolonizing medicine. And decolonizing medicine is a really, it comes out of post-colonial ways of knowing, post-colonial studies, post-colonial literature, and it's looking at this universalizing colonial enterprise of science with a capital S and how it excludes everything else. And to decolonize science allows us to open up to other ways of knowing. So if for thousands of years people have taken pulse to diagnose your illness, how do we study that if it doesn't fit into a randomized control trial? So those are some of the questions that I was also asking on the side. And I found decolonizing medicine as a really powerful framework. And I brought all of this to another I brought all of this to my main research interest when I became a postdoc, which was looking at peripheral sensation. So I had these, interests in neuroscience and also interests in acupuncture, this relationship between acupuncture and neuroscience, but really looking at the body beyond the brain. So if you go to any neuroscience lab, a lot of the questions are about memory, are about dementia, are about learning, about language, and all that centers on the mind in the skull. But we know that nerves innervate the rest of the body. But we don't really know what they do, and we don't really know the full ecology of nerve cells. So for instance, Merkel cells that don't have axons and dendrites, they also transmit mechanosensory input. So what does it mean to explore the body with all these other nerves and neurons that again, in neuroscience, very few people are starting to investigate. There are few people who are asking these questions about physiology because they're hard questions, because they deal with subjects and subjectivity. So that was this expansive moment where I was bringing this decolonizing science into neuroscience and then into my postdoc, which was an interdisciplinary postdoc in society and neuroscience. So that was at Columbia University, and it was called the Presidential Scholars in Society and Neuroscience, which was very experimental at the time and was trying to bring scholars in the humanities in conversation with scholars in the sciences. And that was exactly what we did. And that was also a space for me to think about what my interest as a filmmaker could do. Because when I entered a new space, I would learn about people's work by making a video for them. I would go into a clinic or I would go into a lab and say, do you need this video made? And I would use filmmaking as a kind of form of ethnography to collect data, but then also to return as a gift to the people that I was working with. So that was a kind of social bond that I used. And so in that time, I was also writing a book about the history of meridians and nerves, doing this long comparative history between acupuncture and acupuncture atlases and illustrations of nerves and nervous systems and how they both deal with invisible qualities of the body. And I was also making these videos, and I was in two labs. I was in a touch lab, and I was also mentored by a PI who led a mitochondria lab, which I can talk more about later. But again, this is an effort of trying to triangulate between the sciences, the humanities, and with medicine through an applied approach of a framework that I always held close to my own intellectual core, which was science technology studies. So in that time, just some of the things that I just did, I just made a bunch of videos. I was also closely collaborating with a history of science lab that was dealing with early modern reconstructions of material properties and recipes and practices. They were interested in questions of craft and craftspeople. So these are people who are excluded out of narratives of science. The Royal Society was formed in the 19th century, but they basically appropriated a bunch of practices that craftspeople and artisans were already doing, experimenting with pigments, with chemistry, doing early chemistry, and also creating medical recipes in the home and in their craft space, and appropriating that and bringing that into this elevated space of science for the elite. So a lot of the early modernists who I talked to were also, again, interested in this decolonizing elitist science and looking at people on the ground doing that kind of work. So I worked closely with some of the postdocs in that lab. It was called the Making and Knowing Lab. And I also started a side project looking at Buddhist medicine in Philadelphia. And this was in collaboration with a philologist and medievalist and also a scholar of Buddhist medicine who was looking at the relationship between Buddhism as this pan-Asian approach to, or this pan-Asian practice, and looking at it across an Asian diaspora in Philly. So that's one thing that I was just trying to absorb and learn as much as I could, while again keeping to this core of my own interests in other ways of knowing and of decolonizing science. Even though I still do love science, I think it's cool. I think it's awesome. And one other project I was really proud of, and I was, again, using media as a space to experiment with, was I started this interdisciplinary conversation called Metaphors of the Mind, and it was a series of conversations with neuroscientists, with physiologists, with writers, historians, and scholars of performing arts. And each episode we tried to we tried to interrogate neuroscience based on the metaphors that it used to talk about abstract processes in the body. So, for instance, what is the tension, or what are some of the metaphors that we use to think about pain, to think about language, to think about fear, and how are these social narratives, how are they folded into the science that is practiced and the scientific questions that are asked. So each episode kind of unfolds, and it was this kind of baby of a project that I allowed to blossom while I was a postdoc. And this is just to say that all of these things lead up to Rice. So what am I doing at Rice now? I'm still researching and finishing up my manuscript on this long history of meridians and neurophysiology and representations of acupuncture atlases and territories of sensation. So I still have that book project going on, but I'm also trying to engage with these different kinds of conversations in different kinds of spaces. And in thinking about between science, the humanities, and medicine, these all intersect in the courses that I teach. So when I came to Rice, I started teaching this class on the history of sensation. It's a global and longue durée look at the history of neuroscience and physiology. It's a longue durée look at the history of neuroscience and psychology. And I also last semester taught a FWIS class on the history of poisons, again looking at a global and comparative case of drugs and the things that kill us and cure us. And another class that I've struggled with and come to love is this class called The Body and Global Histories of Medicine. So here we approach the body everywhere, across all places and all times, and it looks at Islamic medicine, it looks at Unani medicine, Ayurveda, it looks at Chinese medicine, pulse taking, it looks at humoral medicine, which is the predecessor of Unani medicine, and we engage with all of these different approaches through post-colonial studies. So this is an ambitious class, it's an advanced class, and it's a class that I offer every other year. And I also teach the intro class to medical humanities, where we think about ethics and how to not be a bad doctor, and the kinds of critical thinking tools you will need in order to, again, think outside of the box to make the kind of change that you want to see, and ask the kinds of questions that people aren't given the space or the opportunity to ask over time, because we're always changing over time, and our answers to these questions are also going to change as we evolve. So just the last couple of things in terms of some projects at Rice that are developing is I also run the Medicine, Race, Democracy Lab, and this is a space that so the Medicine, Race, Democracy Lab has a series of research clusters that look at medicine beyond the TMC. So healthcare and health community clinics at temples and acupuncture clinics and Ayurveda wellness centers, and we compare different narratives of health and wellness across these different spaces. And part of the Medicine, Race, Democracy Lab is a podcast lab called the Metastasis Podcast. And this was something that I started last year during COVID, when a bunch of students had lost their internships, and we were trying to figure out ways to support them to both do research and also produce stories about health and disease. And Metastasis, it started off as a cancer podcast, and now it's inclusive in thinking about different experiences with certain kinds of disorders. So our season 3 is going to include episodes about nightmares and sleep paralysis, and we're also looking at intersex rights and transgender health. And some future episodes also include reviews of cancer experiences in North India, and also, what was another episode? Another episode is about smell and about psychedelics and mental health. So those are some of the episodes we're thinking through right now. Where does this leave you? Where does this leave me? What are some of the tips that I could potentially suggest looking back at the first day of college, where I didn't know that any of this was possible and I had no role models to show me what was possible? Some of the tips are, your plans are rough guides. If you think that your major is all-consuming, you know that it's not. And plans are also scalar. So whatever you plan in the next 2 weeks, in the month, in the semester, in the 4 years that you're here at Rice, those plans can change, but also give yourself space to be flexible, to explore new things, which is my second tip, and that is to honor your interests. If you've always loved, ice dancing, or if you've always been into, I don't know, like forestry, and you're a linguistics major maybe, you can find ways to bridge them together. I know people who like to keep their interests far, far apart, but if there are ways to bring your interests together, that will make you unique to yourself, and that you can, again, find ways and spaces that will help you thrive as long as you give yourself the permission to explore those interests and to keep those interests. And it's okay if some of them fall away, some of them come back. It's totally fine. But keep that in mind. What do you want to do? What are some of the questions that you don't have space to ask, and how do you want to ask that? And finally, the last tip is to keep your curiosity close to you. That will allow you to be open to different ideas that probably are completely foreign and completely contradict what you've known or thought about growing up. So be curious about the world and be open to ways of approaching perspectives that are completely different from your own. And again, that will allow other people to make space for you to be yourself and for you to just have deeper relationships with people who might be strangers now and friends later. So good luck, and I hope that you have a fabulous first semester in spite of everything. Thanks.
Hello, my name is Rafael Verduzco. I'm a professor in chemical and biomolecular engineering here at Rice. 24 years ago, I was an incoming freshman, just like many of you, and I was really excited about getting started here at Rice. Since then, I graduated. I majored in chemical engineering. I went and got my PhD, and then I've returned, and I'm now a professor here at Rice. So what I thought I'd do today is tell you a little bit about some of the activities that go on at Rice outside of the classroom, and give you some advice on making the most of your experience here at Rice. So many of you may know that there's a lot that goes on at Rice outside of the classroom. Some of these examples I have on this slide were just posted today or this week. They're discoveries, findings, or just activities that are going on that you can learn about and get involved with. Just to give you an example, so I lead a lab here at Rice in chemical engineering. These are a couple of pictures of my lab in Keck Hall, and photos of my graduate students and some of the postdoctoral researchers. I also have a lot of undergrads that come to the lab and work in the lab. So these researchers and graduate students that you see here, every day they come to the lab, they're working on a research project, trying to solve problems and challenges that'll benefit, that'll be reported to other scientists and sometimes to the general public, and will hopefully benefit a lot of people. So you may wonder, what kind of problems do you work on? How do you find a problem? Are these challenges really something that I can get involved with, and are they interesting? So I want to highlight one example of a project that my group's been working on for about a year now. So just about a year ago, the Defense Advanced Research Projects Agency put out a call for ideas. They wanted a team to come up with a sensor that could sample air, detect the presence of SARS-CoV-2 within 5 minutes, only respond to the presence of SARS-CoV-2 and no other viruses, no other coronaviruses, no other particles that may be in aerosols. And then, of course, do so at low cost and ideally with a disposable system that could be distributed everywhere. So this is quite a challenge, and this is probably not something that any one group or professor or person could handle on their own. But luckily, we have a lot of professors and groups at Rice with different expertise. And so I spoke with Professor Jane Tao in biosciences, Pedro Alvarez in civil and environmental engineering. And we came up with an idea that we thought could address exactly what DARPA was looking for. So this was, what I'm showing here is an early sketch of our idea. And so what we thought could work for this type of sensor is to first design a thin polymer layer. We call this a molecularly imprinted layer that'll have pockets that can bind just to the SARS-CoV-2 virus. Then this film will be incorporated into a low-cost electronic device, and then that can be incorporated into a larger device with onboard power and wireless capabilities, so that then you can have these sensors that are distributed through the household, sampling the air for the presence of coronavirus, or even in wastewater, and telling you whether or not there's SARS-CoV-2 in the vicinity. And so, just to tell you a little bit more about the part that I'm working on, it's the actual sensor. The sensor is an organic electrochemical transistor, and I show it here on this slide. It's actually a pretty simple electronic device. So it involves 3 electrodes: a source electrode, a drain electrode, and a gate electrode. And in between the source and drain electrode is a semiconductive polymer. So all this device does is it applies a voltage across that source and drain and measures a current. But then once that gate voltage turns on, it drives electrolyte into that semiconductive polymer layer, and it reduces the conductivity, and you see a drop in conductivity. And so this plot shows a typical response of a device like this, where that source drain current decreases as the gate voltage increases. The reason that these could be used as a sensor is because anything that binds to that gate electrode will affect the effective voltage and affect that conductivity in that semiconductive polymer channel. So if we have a layer that's selective to the coronavirus or the spike protein, and we happen to bind to one of these on the gate electrode, we'll see a significant drop in that source drain current. So when we started working on this project, we also needed a low-cost way to make these and make sensors that were disposable, because you want to be able to test and replace the sensors quickly and cheaply if needed. And this is a schematic of some of the sensors that we were made, mostly handmade in the lab by a variety of students, researchers, and also undergrads working on this project. We can then take this completed sensor, immerse it in our liquid for testing, and test for the presence of our biomolecule, or SARS-CoV-2. Since then, we've done further engineering and miniaturized them further, and this is an example of the sensors we're using now. They're about the size of a quarter. There's a little well that'll hold your liquid sample, and you can just clip leads onto either end and measure the current to see whether or not there's a response. So the next step in building our sensor was to find a way to bind specifically to the SARS-CoV-2 virus. The SARS-CoV-2 virus has a number of spike proteins on its surface, about 75 per each virus particle. These are the proteins that actually allow it to bind to human cells and enter cells and replicate. And so luckily for us, there had already been a lot of work over the past year in identifying proteins that would bind to the spike protein. There's been a lot of work in engineering proteins that'll detect the SARS-CoV-2 virus and spike protein very specifically, and not respond or bind to any other virus. So we were able to get a sample of this and use it in our sensors. This schematic shows some cryo-EM reconstructions of how these proteins named LCB1 bind to the spike protein. So then we were ready for testing. So we took our organic electrochemical transistors, we introduced LCB1 onto the gate electrode, and this is a schematic of the first computer setup we have. Very low-tech, kind of homemade, right? We used alligator clips and a couple of potentiostats to measure current. But our very first test produced very exciting results. So what I'm showing on the left-hand side is a plot of the source drain current measured as a function of gate voltage for different concentrations of the spike protein added. So we went all the way down to 1 yoctomolar of spike protein. That's 10 to the minus 24 molar, and all the way up to 10 picomolar. And it's hard to tell when I overlay all of these, but what happens from 0 to 10 to the minus 21 molar is there's no response. So below 10 to the minus 21 molar, the sensor doesn't detect the spike protein. But once you get above to 10 to the minus 20 molar or above, all of a sudden there's a large shift in that source drain current. And as more and more spike protein are added to the sensor, you see a larger and larger shift. And so what this tells us is that we can very sensitively detect the presence of spike protein. And not only can we detect it at 10 to the minus 20, we can actually tell you exactly how much spike protein is in that solution, up to at least 10 picomolar. And yeah, this plot shows how we would actually calibrate this sensor response. And you see that We get a baseline response below 10 to the minus 20. From 10 to the minus 20 at higher concentrations, we see just this increasing response of our sensor as reflected by the shift in this source drain current. So we were very excited to find that our sensors worked. What we've done since then is to try to build everything else around it, right? So DARPA also wants aerosol sampling. And so we've teamed up with a startup company that is developing an aerosol sampling device. They call it the Blowfish Sentinel, that can sample air, condense any aerosol particles into a liquid stream that can then be tested by our device. In between this aerosol collector, there's a microfluidics setup that can transfer liquid to our test chamber, and then we can automate this entire device so that we can do testing every 10 or 15 minutes, however long it takes to collect aerosol and run a reading. The device that I show on the left-hand side is really just a concept or a schematic of what we imagine this will eventually look like. Depending on how large these components are, it could be a tabletop device or even maybe a portable handheld device that can constantly sample air in the environment. So I hope that gives you a flavor of just some of the research that goes on at Rice and gives you an idea that the work that we're doing outside of the classroom, it's not just for other scientists. It's not just esoteric content. It's stuff that matters and stuff that's important. And so just to give you some advice to close out my talk, I strongly encourage you to go beyond the classroom and get involved in extracurricular activities. I'm in science and engineering, so I talked about an example in engineering, but you're certainly not limited to that, right? I definitely encourage you to get involved in student government, maybe in getting involved in the paper, in the Rice Thresher. There's a lot of activities going on that you can be a part of as an undergrad. Again, I want to emphasize the work that's going on at Rice is important and relevant. And I think it's something that as you dig through some of the opportunities and activities that are going around on campus, you can get excited about and really want to get involved in. And as an undergraduate student, even if you don't have any prior experience doing research or even have worked before, or worked at a newspaper or anything like that, you can make significant and meaningful contributions to all these activities. Right? As an undergrad, you'll be trained on an aspect of a project, and as you work on it, you learn more and more, and you can make a significant contribution to some of these activities. And last but not least, work is important, classes are important, but of course it's really important that while you're here at Rice is you make friends and have fun. I'm lucky enough that I'm still in touch with my freshman year roommates and suite mates and a lot of other friends that I made at Rice, and that'll be some of the most meaningful things you accomplish while you're a student here, is just meeting other people and connecting with others while you're here. So with that, I'd like to welcome you to Rice again, and I look forward to seeing you on campus this fall.